This is the third video in the AP Biology Review series, and it is about energy and enzymes. So starting with metabolism. Metabolism is the total of all the chemical reactions that take place in cells. There are metabolic pathways, which are the series of steps each metabolic reaction occurs in. Each step is very important, and the ultimate goal of each cell is to be energy efficient. So in the corner there, I have a picture of glycolysis. I know everyone remembers it very fondly. And this is an example of a metabolic pathway. We start with glucose, then through a series of reactions with different enzymes and intermediate molecules, we will end up with pyruvate. So even though it seems like, oh, there's too many steps, why are there so many molecules, this pathway is actually very energy efficient for your cells. There are catabolic pathways, which are where molecules are being broken down. So the energy that is stored in the molecules becomes usable to the cell, and energy is released. So as you can see in the picture, there are molecules bonded together, and when those bonds are broken, energy is released. Then we have the opposite, anabolic pathways, which are how molecules are created. And for example, let's say we're joining amino acids to create a protein, and energy is being used. So as you can see in the picture, we have separate molecules, but when we add energy, we connect them, create bonds between them. A few forms of energy. We have kinetic energy, which is the energy of motion. And an example would be heat or thermal energy, because heat is really measuring the speed of atoms and molecules. Here's a picture of some gas molecules moving around very quickly, form of kinetic energy. Then there is potential energy, which has the potential to do work. And chemical energy is the potential energy of molecules. And it's released when broken down through catabolic pathways like we talked about in the previous slide. Over there, there's a picture of sucrose. Sucrose would have potential energy, specifically chemical energy, because it has the potential to do work when its bonds are broken, because energy will be released when that happens. Here there's a picture basically comparing kinetic and potential energy. The first picture on the left, the cyclist is at the top of the hill. They have potential energy. They have the potential to do work. And as the cyclist is going down the hill, they have kinetic energy, the energy of motion. Thermodynamics is the study of energy transformation. The first law of thermodynamics is that energy can be transferred and transformed, but can never be created or destroyed. So basically, energy is always being conserved. And the ultimate source of energy is the sun, which is why I have that picture there of the sun and plants, because the sun provides light energy for plants to do photosynthesis, create sugars. Animals will then eat plants. Animals will eat other animals that have eaten plants. So basically, as you can see, energy is always being transferred between organisms. The second law of thermodynamics is that all energy transfers and transformations increase entropy in the universe, and entropy is a measure of disorder. So that's just a brief overview of the thermodynamics section of this chapter. Now let's talk about the relationship between stability, free energy, and work. So Gibbs free energy, just wrote as a G, so that's the energy available to a cell that could be used to do work. Delta G is the change in free energy, so we take the final amount of free energy and subtract the initial amount of energy from it. Here's a hill that I have drawn, and there's a boulder at the top of it. At the top of the hill, the boulder is less stable because someone could push it off and it would fall. It has more free energy at the top, And it has a greater ability to do work, because if it's pushed, it will do work and go down the hill. So let's pretend someone pushes it. This is my attempt at an animation. And it's at the bottom and in pieces on the floor. In this state, it is more stable, right? Because the pieces can't really do anything at that stage. There is less free energy. Again, those pieces are not really able to do any work. So again, lower ability to do work. Free energy. So again, the change in free energy is final minus initial. Exergonic reactions are reactions in which the delta G is less than 0. So that means that we're starting the initial amount of free energy is greater than the final amount of free energy. And that means that energy is being released in these reactions. And they are called spontaneous reactions because no outside energy is needed to start them. And an example would be cellular respiration. Cellular respiration has a negative delta G, which means it's exergonic. It starts with more energy than it ends up with because sugar is being broken down. So here, this is what a graph of the free energy levels in an exergonic reaction would look like. As you can see, the reactants start off with more free energy than the products, and energy is released. The opposite would be endergonic reactions. Delta G is greater than 0, which means that the energy is being absorbed, right? Because the final amount is greater than the initial amount of energy. And an example would be making ATP, so adenosine triphosphate, from ADP diphosphate, so that has 2 phosphates, and phosphate. So adding a phosphate to ADP to create ATP would be an example of an endergonic reaction. And as you can see in the picture, this is what the graph of the free energy would look like. It's the reactant amount of free energy is lower than the product, which means that energy must be added. So this would not be a spontaneous reaction because energy needs to be added to start them. Then we have coupled reactions, which means that energy from one reaction is used to power the next. So an exergonic process would drive an endergonic one. And an example would be ATP hydrolysis, which means that breaking apart of ATP would power other reactions by phosphorylation. So phosphorylation is basically transferring that phosphate group, because remember, ATP will become ADP and P, and so that P would is a phosphate group that would be transferred to another molecule. So here's a graph. If we should look at it in portions. So as you can see in the first portion, glucose being broken down is powered by the creation of ATP from ADP and P. So you can see that the arrow from glucose to carbon dioxide, water, and heat is touched by the arrow going the opposite direction from the creation of ATP. So you can see that those two kind of balance each other. And the same is true for the opposite reaction. ATP being broken down could help power the formation of polypeptide from amino acids. So these reactions would be called coupled, because one powers the next. Next we have enzymes. So an important thing to talk about before enzymes would be activation energy. Activation energy is the amount of energy needed to start a reaction. So as you can see in the picture, start with reactants, and then there's this large hill basically that needs to be overcome in order for the reaction to begin. Catalysts are things that lower activation energy. So they make the reaction happen faster. Enzymes are examples of catalysts. They are specifically proteins with very specific functions. So each enzyme has a very specific role, and it will only would only help lower the activation energy of certain reactions. So remember, we talked about in the last video, proteins have a very specific shape. The way that it's folded has a lot to do with its function, and enzymes are proteins, so that will apply to them as well. And cofactors are basically non-protein helpers that help enzymes do their job. So again, let's take a look at the picture. You can see the activation energy is very high, but when you add a catalyst, the activation energy will decrease. The hill is not as tall, which means that it will have an easier chance of getting to the top of that hill and starting the reaction. So it makes the reaction happen faster. More about enzymes. There is a substrate, which is the substance or substances that the enzyme will act on. The active site is the region where the substrate binds to the enzyme. And we'll talk about induced fit in a minute, but let's look at the picture really quickly. As you can see, we have an enzyme that I drew. I mean, they're not actually going to look like that. And there's a little hole basically where the substrate will fit into the enzyme, and that hole is the active site, and the substrate is the thing that will that the enzyme will act on, and that will go into that active site. So induced fit is when the substrate enters, the active site will slightly change to better fit around the substrate. So a lot of times people used to say like the enzymes and substrates are like lock and key, but that's not really true, because in reality the lock will kind of change shape for the key to fit in better, so to say. So basically the active site is not something rigid. It's something that will bend to better have a better grip basically on the substrate. And the enzyme substrate complex is when the substrate is in the enzyme. Both of them together make up the enzyme substrate complex. So what happens after the substrate is binded? The enzyme will speed up the reaction. Substrates will be made into products. Products will be released, and the cycle will continue. So basically, new substrate molecules can bind to the enzyme, and the enzyme is reusable. It'll keep doing its job. So here's a picture that kind of summarizes that. Again, we have a substrate binding to an active site, and this picture does a good job of showing how the active site originally is not the perfect shape of the substrate, but there's induced fit, so the enzyme will change shape slightly to bind to the substrate better, and then the substrate will be changed into products, and the products will be released, and the active site of the enzyme will be empty again, and a new substrate molecule will bind to it. So the environment will affect enzyme activity. Temperature, up to a certain point, an increase in temperature increases the reaction rate, and this is because substrates are colliding with the active sites more. So again, like we talked about earlier, heat and temperature affects how fast the molecules are moving. So up to a certain point, increasing the temperature will increase the speed at which the substrate molecules are moving, which means that they'll collide with the active sites more, so the reaction rate will be increased. However, each enzyme has an optimal temperature. So as you can see in this picture, there's an optimal temperature. That's when the rate of reaction is at its peak. And again, I said that up to a certain point, an increase in temperature increases reaction rate. But as you can see in the picture, after about 40 degrees Celsius, the reaction rate will decrease pretty quickly until about 60 degrees Celsius, where it won't even happen anymore. And this is because enzymes, again, are proteins, and if the temperature is just too high, they'll denature and no longer be able to do their jobs. pH also affects enzyme activity. Again, each enzyme has an optimal pH, just like it has an optimal temperature. And once again, it can denature if put in a more acidic environment. But just to reiterate, each enzyme is very specific for what it does. So different enzymes will have different optimal temperatures, different optimal pHs. And this is another picture showing pH. You can see there's an optimal one where reaction rate is highest, and if we lower it, the reaction will decrease. And if you lower it enough, it won't even happen. And the same goes for the other side. If you make it too high, it won't happen. Enzyme inhibitors. So these act to limit the number of reactions occurring. And again, we need to keep in mind that our cells want to be metabolically efficient. So we don't want reactions to be occurring that are not necessary, because that would just be wasting energy and resources. So competitive inhibition is when another molecule is in the active site, and that doesn't allow other substrates to bind to it. Non-competitive inhibition is when the active site is deformed because a molecule binds to the enzyme in another place. And the molecule binding to an enzyme in another place actually changes the shape of the active site. So here's a picture to show you the difference between the two. Again, the first one, the inhibitor is actually in the active site, so you can see the substrate can no longer go there. Non-competitive, the inhibitor binded to another part of the enzyme, but that changes the shape of the active site, so the substrate can no longer fit in the active site. So this is more regulation, allosteric regulation. And allosteric basically means a change in shape. So the enzyme's function at one site is affected by the binding of a molecule to the other site. So the non-competitive inhibition we talked about would be an example of allosteric regulation as well. So again, the binding of the molecule at one spot changes the enzyme shape at another. There are allosteric activators, which basically stabilize the enzyme in active form, and allosteric inhibitors, which do the opposite. They stabilize the enzyme in inactive form. Cooperativity, sorry, I'm not sure if I'm saying that right, is a type of allosteric regulation. So binding of one substrate to an active site stabilizes all the active sites into active conformation. So let's take a look at the pictures. Allosteric inhibition. This is kind of like the non-competitive one again that we talked about. The inhibitor binds to a spot on the enzyme, the allosteric site. It's not the active site, but it'll change the shape of the active site so that the substrate can no longer bind. Allosteric activation is the opposite. The activator binding to a site on the enzyme changes the active site so that it can bind to a substrate. And then below, that's cooperativity. Sorry, again, I don't think I'm saying that right. The substrate will bind to one of the active sites, and it'll change all the other active sites on the enzyme to be in the active conformation. And that would be especially important for enzymes that are made of multiple polypeptides, so they'll have several active sites. Feedback inhibition is when the end product inhibits its own creation. So again, we want to be metabolically efficient so that it'll stop the cell from wasting energy and resources making too much of a substance it already has enough of. And an example would be isoleucine synthesis. Isoleucine is an amino acid. Threonine is the initial substrate, and it'll bind to an enzyme. Then several intermediate molecules are created in several steps, different enzymes, and the end product, isoleucine, will be created. Then the isoleucine will bind to an allosteric site on the original enzyme used, changing the enzyme's conformation so it can no longer bind to threonine. And as the isoleucine gets used up by the cell, it'll leave the enzyme and threonine will bind to the enzyme again. So here's a picture that basically summarizes all of that up. Again, you can see threonine, series of intermediate substrates and enzymes, and then we have isoleucine. So large concentrations of substrate will mean that the enzyme, it'll have a higher chance of coming in contact with the enzyme. So when there's a lot of isoleucine, the isoleucine will come into contact with the enzyme and will bind in the allosteric site, which will basically stop the enzyme from doing this threonine pathway, and it'll stop threonine from being able to bind to that enzyme. But when the isoleucine gets used up, the isoleucine will leave the allosteric site, and the active site will become available to threonine again. So again, isoleucine is stopping its own creation because it would be a waste for the cell to create more isoleucine than necessary. And that's it for this video. Thank you so much for watching, and please subscribe if you'd like to see more videos.